Everyone, welcome to Rakesh Sir Lectures, and today we are going to see about glycolysis. And as earlier videos I have discussed about the basic of glycolysis, and we have seen that from how the glucose is reaches to the cell. So let's assume this is cell, right? This is the cell membrane, and this is one cell. And we have seen that from systemic circulations how the glucose reaches inside the cells. So let's assume these are the glucose, and we have discussed already how through the diffusion the glucose from the capillaries it will come into the space, that is interstitial space between the cells and the capillaries. So these are the glucose molecules which will be just permeable, and the concentration of glucose inside the capillaries and interstitial space is same, right? Because it is freely permeable. Glucose is freely permeable from the capillaries to the interstitial space. But the concentration of glucose inside the cells and interstitial space is not the same. So concentration here is less and here is more. And we also have seen that from here, with the help of transporter, glucose transporter, and glucose will move from interstitial space to inside the cell, intracellular space, right? So, and we have also seen there are different types of transporter, right? There are different types of transporter which helps glucose to move from extracellular to intracellular, because the membrane present here, cell membrane is not permeable for the glucose, because the size of the glucose is larger. That means it cannot pass through the cell membrane. Okay, so we need a special type of transporter. So we have seen. So today lectures mainly focuses the steps, the different steps present in the glycolysis, right? And what are the importance of each steps, right? And how each step is regulated, right? So in detail we are going to say about steps of glycolysis in this lectures, right? So before I start these lectures, I want to highlight few things, like what is glycolysis? What are the importance of glycolysis? So once we know the importance of glycolysis, then we'll get little motivations and satisfactions to know the different steps involved in the glycolysis, right? So before going to the detailed steps, let's define what is glycolysis. So how do you define glycolysis? Yeah. So the glycolysis can be defined as it is the metabolic pathway in which the oxidation of glucose or glucose is oxidized into the pyruvate, right? So or lactate. So it is the oxidation of glucose, right? Once the glucose from here it reaches here, so glucose will convert into, with the several series of reactions, glucose can be converted into pyruvate, and in some conditions it can be converted into lactate, right? So the conversion of glucose into pyruvate in aerobic conditions, or in absence of oxygen or absence of mitochondria, it can convert into lactate or lactic acid. So this is known as glycolysis. So it is a metabolic pathway in which glucose is oxidized into pyruvate or lactate. It's a simple definitions, right? So now we are going to see the importance. What is the importance? So there are several importance. The provision of these reactions is just to provide the cells energy, right? So it will emit energy, ATP. So the provision of this glycolysis is to provide energy. So first importance is it provides energy, right? Second, so what is the use of this energy, importance of getting energy, right? So first is, in our skeletal muscles, you have seen that when you are in heavy exercise, the energy requirement the energy requirement by the cells is not enough, right? So the oxygen supply in the anaerobic in high exercise will be considered as anaerobic phase, right? Because oxygen demand is not fulfilled as compared to the energy demand, right? So that means in during heavy exercises in skeletal muscles, it will be act as anaerobic phase. So it will be act as anaerobic conditions or during ischemic conditions in skeletal muscles. During ischemic condition in skeletal muscles, this glucose will be converted into lactate, right? Glucose will be converted into lactate, and during these reactions ATP is produced and skeletal muscles can easily survive. So even in ischemic conditions skeletal muscles can survive because of this glycolysis, anaerobic glycolysis. Okay, so here I can put as importance for skeletal muscles in ischemic conditions as well as during heavy exercise, but these things will not happen in cardiac muscles. These things will not happen in cardiac muscles because cardiac muscles are mainly manufactured in such a way or work in such a way that they will only run aerobic glycolysis. Okay, so in aerobic glycolysis only pyruvate is formed, and after a period of ischemic conditions, this reaction will stop, glycolysis will stop, and the energy production will stop, and or it will decrease, so that it will lead to different kind of ischemic attack in cardiac muscles. Okay, so I will tell you why this will happens in anaerobic conditions, like when the absence of oxygen, cell will survive, and in that this skeletal muscles will survive, but cardiac muscles will not. Right? What is this condition? Why this glycolysis will stop? Glycolysis will stop, and why in this anaerobic conditions glycolysis will run? The major thing is, later in detail of course I will talk, but here you need to remember is when the glucose is converted into pyruvate, of course ATP is released. In addition to ATP, it converts NAD into NADH. Okay, NADH. So this NAD plus is converted into NADH. That is very, very important. That means this during this glycolysis, NAD plus is utilized, right? This is utilized, and this NADH in presence of mitochondria and TCA, this ultimately this pyruvate should run into TCA cycles, and ultimately again this electron transport chain, right? ETC. And then this NAD should get oxidized and produce energy, right? But I am talking about the condition ischemic conditions in cardiac muscles. Ischemic. First, let us talk in detail about first skeletal muscles. So in anaerobic absence of oxygen, what will happen? This pyruvate, once this is converted in pyruvate, in anaerobic conditions, it will not go to TC and ATC. So what this pyruvate will do? It will convert to lactate. But why it is converted to lactate? Is there any significance, clinical significance? Because there is no production of ATP. Then why simply it should lactate should produce here? The major reason is, once this pyruvate is converting into lactate, here is, already there is NADH produced. During this cycle, NADH is produced, and this NADH is utilized here. That means NADH will convert here into NAD plus. NADH is utilized. Okay, this NADH is utilized, and this once it is utilized, this now it converted to NAD plus, and this NAD plus again it will go here, and it will make this glucose again to convert into this way. So that means once anaerobic glycolysis, this pyruvate will convert into lactate. Same time there will be conversion of NADH, because this is trapped. This NAD, we need enough amount of NAD plus to run this cycle, right? To run this cycle, glucose to pyruvate, we need enough amount of NAD plus. And this NAD plus, if it is only pyruvate, then NAD plus is trapped in the form of NADH. Right, and with a short period of time only, we not have enough amount of NAD plus to run this glycolysis. So we need to get NAD plus. From where will we get it? Just convert pyruvate into lactate. This NADH is utilized, hydrogen is removed, NAD, you will get NAD plus, right? And this NAD plus will help at least that cell to convert glucose into pyruvate. At least you will get ATP. This ATP. At least you will get ATP, and that ATP at least help for that certain period of time for the survival of the cells, right? ATP is nothing but energy. So that means in skeletal muscles, this will be happening. Lactate production is happening. That means NADH is here, production of NADH is there, and this NADH is utilized here, and NAD plus is formed, and this NAD plus is again going back and helping these cycles to not to stop, to run it, right? To run it. So in skeletal muscle, this glycolysis will not stop. It will run it because it will all converting into lactate and NAD plus is free. Am I right? This most of the students will get confused, right? Same thing we'll see for cardiac muscles. In cardiac muscles, what will happen? I told they will not go for lactate, anaerobic. They will prefer, their cells are meant for aerobic glycolysis. So what will happen there is, for when there will be ischemic attack or ischemia, so that what will happen? Glucose will convert into pyruvate. Okay, glucose will convert into pyruvate. Of course, for a short period of time, it will convert, like it will provide ATP and it will provide NADH. But this when the all NAD plus is utilized for NADH, that means there is no now NAD plus, right? So when there will be no NAD plus, so this conversion of glucose to pyruvate will stop. So that means this reaction will stop. That means at least this ATP production, few amount of ATP which is getting produced, that will get a stop. That means in cardiac muscles they will have problem. Cell will start degenerating, cell and dying off, right? So cardiac muscle cannot survive, right? So anaerobic glycolysis is very, very important. Are you getting it? To run the glycolysis, at least to run the glycolysis, anaerobic glycolysis is important. To run the glycolysis, this, right? So, okay, one more importance is there in cancer. In cancer, do you know about tumor cell? It is rapid growing cells, right? And in tumor cells, you find that it is not just rapid growing. How it is, all the glucose it is utilized here. If this is the tumor cells, the glucose what is uptake by the tumor cell, the rate of glycolysis is so high. See here the clinical significance. The rate of glycolysis as compared to normal cells, tumor cell has more. So once the tumor cell is converted from glucose to pyruvate, that means more amount of pyruvate will be found because the rate of glycolysis is more. But the rate of uptake of this pyruvate to the TCA cycle is less as compared to the rate of production. So rate of glycolysis is more, but the rate of TCA cycle is again the normal, what is as compared to normal individuals. Okay. Because the normal number of mitochondria remains the same, right? So TCA occurs in mitochondria. So here the production of pyruvate is more, but the utilization of pyruvate in TCA cycle is less. That means there will be accumulation of pyruvate inside the tumor cell. What will happen? That accumulation of pyruvate, it will go to the anaerobic glycolysis, and there lactate is formed, right? And this lactate or lactic acids will accumulate inside the tumor or nearby tumor cells, right? And the tumor cells will produce lactic acidosis or some acidosis, right? Local acidosis in that area. So once that local acidosis will be there, you will find some other clinical signs and symptoms related to that. So it's very important to know about in the cancer also. It is very. There are several other also, but at least main things you remember. It is meant for provision to provide energy. So when we are going to in detail about this glycolysis, we will be saying that we'll more concerned about the ATP. How much ATP we will gain at the end of the glycolysis. Okay. So we have seen this much importance, right? So we have to show gratitude to the inventor who has invented these biochemical reactions, right? So the inventor are we have embedded Meyerhof and Parnas. There are three inventors which invented this glycolysis reactions, and based on these three names, we will call it as EMP pathway. first starting letter, we'll call it as EMP pathway, right? Am I clear? So glycolysis pathway, or it is also known as EMP pathway, and one more name is there. The enzyme which is required to convert glucose into pyruvate. There are several enzymes, series of enzymes. That all enzymes will present in the which part of the cell? It will present in the cytosol of the cell, plasma of the cells. So it is also known as cytosolic pathway. Because later I will tell you this pyruvate will be enter inside the mitochondria and then tissue will occur. So this is the mitochondrial pathway, right? So but this glycolysis will occur in the cytosol, so it is called as cytosolic pathway. Am I clear? Hope I am clear. So now we have seen the different names and definitions. We have seen the importance of this glycolysis. Then we have seen about what are the different names, inventor, and where the enzymes are located. So now we are going in detail steps of each step. You are getting it? So I erase this and we'll go in detail about each steps and we'll see how this has been regulated, right? So I hope you guys are knowing about a brief of glycolysis. So first things, with the help of transporter or whatever things, if it is liver cells, liver cell has GLUT2 receptor, GLUT2 transporter, which can freely able to 2 ways. GLUT2 is 2-way. So if it is liver cells, glucose can enter and exit out. It's freely easily, because it is free, this liver cell or hepatic cells is freely permeable to glucose, right? If you take about other cells in our body, like adipose tissue, renal tissue, and all other cells, they have some different type of transporter which helps that to move inside, right? Muscles, we have GLUT4, like in ocular tissue, if you are seeing about cornea, conjunctiva, sclera, ciliary body, iris, choroid, retinal cells, they all follow some transporter so that the glucose can enter inside to the cell, right? So with the help of transporter, let's assume the cells take glucose inside the cells, right? So let's assume this is liver cells and this glucose enter inside to the cells. So I'm just drawing it here. So glucose enter inside the cells, and as you know, glucose is how many carbon molecules? 6 carbon molecules. How many carbon molecules? 6. So I'll draw here 6, 1, 6. So this is glucose, right? So once the glucose enter inside the cells, the first action by the cell to the glucose is trap it, because cell don't want to lose this again back, because if this is glucose is so nutty that it can, if you not trap it, then it will it may that it can move it back, right? So first things the cell should do is they will trap the glucose, and after that the cell will decide which metabolic pathway that product should go, that glucose should go. Either it should go glycolysis, or either it should go gluconeogenesis, or either it should follow the glycogenesis. Then either it should go to uronic acid pathway. Let whatever the metabolic pathway is there, first the cell should trap this glucose. And how will trap it? If someone will come inside your room, how will you trap it? Just tie them, right? Tie with chain or tie with something. Just tie them so that they cannot move out, right? So same thing cell will do for the glucose, right? So what they will do is this glucose will, what cell will do is the glucose will be tied with, literally it will tie, glucose will tie with phosphate. They will tie with the chain of phosphate. So what they will do is the glucose will, this glucose will be at the 6 carbon atom, they will tie a chain of phosphate. phosphate. Now this product will become phosphorylated. That means charged molecules. Now the glucose becomes charged molecule, and also it becomes larger in size. Right. So now, as it becomes larger in size and mainly highly polar compound, highly charged compound, highly polar compound. Now charged molecule cannot be passed through the membrane. So now it got trapped. Now once the molecules become polar, it cannot go out of the cells. That means it becomes trapped now. So anyway, once the cell, this product becomes trapped, now the cell has to decide what to do. But let's first see in detail about these steps. Let me draw with this red color, because this step is very, very important. Because in this step, this glucose decide now, glucose will commit now. Commit means the sense it cannot revert it back. This is irreversible step. This is irreversible step. Sorry. This is irreversible step. Right. So now cell glucose will commit to be inside the cell. Right. In this step, glucose will be commit to be inside the cell. And what is this product name? Glucose 6-phosphate. Right. At 6th position, phosphate is attached. But I want to tell few more things about this step. First, I want to highlight from where this phosphate is come. From where this phosphate is come, and what are the enzymes which makes this glucose to convert into glucose 6-phosphate. So let's assume here this phosphate will come from ATP. Let's ATP triphosphate, right? It will convert into diphosphate. That means one phosphate is released. One phosphate is released, and which got attached at this point of time, right? Which got attached here. And this will be catalyzed by a mineral magnesium. Catalyzed by a mineral magnesium. So what we can do here is, and this enzyme which is responsible for this is hexokinase or glucokinase. I love to mention few more points on this enzyme because this is very, very important enzyme. What I want to mention is this hexokinase. Of course, kinase. Kinase means wherever the in biochemical reactions, kinase, what will come? That kinase indicate it is adding phosphate in the substrate. Okay, it is adding phosphate in the substrate. So this both enzyme, of course, it will add phosphate by dephosphorylizing the ATP. Right. So this is one thing, common things, right? Other thing, if you want to track the activity of the enzymes, we have one curve. This curve, this is a curve, and in this curve we will be seeing like okay, and so this is one enzyme activity. Here is the concentration of substrate, or we can concentration of substrate. So we can keep here concentration of substrate in the sense here it is glucose, right? Substrate, here it will be glucose. And one more enzymes, I will draw the graph how it will be. So I will put here okay. So let's assume in horizontal we have seen the concentration of glucose, and in vertical we will see the reaction velocity of the reaction velocity, or it is also called as, you can think of it as the performance of the enzyme. It is the performance of the enzyme. So first I'll write whatever you have seen this curve. This is the hexokinase activity, and this is the condition of, this is for glucokinase activity. Okay. Now see here everyone. See the it will be indicated first as Km. The performance. You just see. I'll tell what is Km. Km is the concentration of glucose, the concentration of substrate or concentration of glucose at which the reaction velocity is half. So after this So you see for hexokinase first. So in hexokinase, you are seeing that once you increase little concentration of glucose only, little concentration, this is getting half, right? This is getting almost half. After half, it is getting a straight curve, right? So km4 or km is low. Km is low for hexokinases. If we talk about affinity of this hexokinase, or since affinity of this hexokinase toward this glucose is more. Affinity is more because if little glucose only, this hexokinase is reaction is more. Reaction velocity is more. That means it is very sensitive to glucose. Hexokinase is very sensitive to glucose, right? But it cannot take more amount of glucose. If you keep increasing the glucose, at certain period of time it cannot take, the reaction velocity will remain same, right? So it cannot uptake, or it cannot convert more amount of glucose into glucose 6-phosphate, because after once you increase the concentration, their rate of reaction remains same, right? So Km is at what point? So at low only, at low concentration only, it become half saturated, right? So if we talk about the affinity or sensitivity, affinity is more, because in less only it is reaction is more, right? So this is affinity is more, increases, affinity is more. Similar things will compare with glucokinase. If you keep increasing the concentration of glucose after so long, okay, after so high concentration, then only it become half saturated, right? Let's assume this is the half, right? Halfway. So if you keep increasing the concentration of glucose, then maybe after so high concentration, it will become half saturated. That means Km is more. Km is more for glucokinase. Km is high. Km is high. But what about affinity? Sensitive toward glucose. If you give little concentration, this will not go more. You are seeing it? So it will not go more. If you keep increasing, then the reaction velocity will be more. Let me draw this in this way. This curve actually theoretically it is not correct. So let me draw in this way. Okay, yeah, this is now the correct of this. Here it is glucokinase. Now you see, if I increase the concentration, the velocity is still less. If I increase there, it is still less, right? But at this point, if you see, their reaction velocity is, if I talk about this point, at this point, right, same level I am taking. At this point, glucokinase reaction velocity is less, but hexokinase is more. That means at the Km concentration of glucose at which this reaction velocity or performance of the enzyme. So performance of the enzyme at the same concentration, hexokinase has more, but glucokinase is less. That means even in low glucose level, hexokinase can perform well, but glucokinase cannot, right? But if you give the high concentration of glucose, hexokinase fail, but glucokinase can run, right? It can utilize all the glucose to convert into glucose 6-phosphate. Am I clear? I hope I am clear with this. If you have any doubt, please comment me. Right? I'll clear it out. So this is one thing. Other thing is, other difference is, this hexokinase will be present in all our part of the body, like adipose tissue, muscles, skeletal muscles, smooth muscles, and other connective tissue cells, right? And almost all part of the body. Glucokinase will present especially in liver, right? And even hexokinase also present in liver in female, but glucokinase is more specific to liver, and hexokinase is present extrahepatic, right? Extra liver. So this is one. So we have seen about this reaction velocity also, right? Now I want to highlight about a few more things about the regulation of the enzymes. See the more interesting concept about the regulation of the enzyme. What will happen is, glucose is converting into glucose 6-phosphate, right? with the help of the same enzyme. But after some times, when you are in fed state, let's just assume like when you have a heavy meals, right? And just after the heavy meals, high amount of from the GIT, high amount of glucose is absorbed and it will go to liver. So when this will go to when high amount of glucose is coming to livers, high concentration of glucose is coming to livers, some of the glucose is utilized, they will run through the glycolysis, Krebs cycles and electron transport chains, and it produces ATP energy. So let's assume what the requirement of the ATP is fulfilled. Still the glucose is coming because you had a heavy meal. So that remaining glucose is converted into glucose 6-phosphate. From here it will divert toward the glycogenesis. So it is stored as a form of glycogen. Right. So once let's assume you have enough amount of ATP, you have enough amount of glycogen produced, and it is stored. Right. Now still the glucose is coming. What you will do? Do you want that glucose to be here, or you want to let it go out? Because of course you can convert it into some other amino acid and protein. So after that the reaction should slow down, because the rate of reactions at the beginning of the glycolysis and at the late phase it will be slow down, right? At the beginning it will be relatively faster. Once there will be enough supply of ATP and glycogen storage will be there, then the rate will be slow down. So what we can do here is once the rate will be slow down, there will be accumulation of glucose 6-phosphate, right? Because already glycogen storage is there, already the forward directions will be less rate as compared to this conversion. So production of glucose 6-phosphate is more as compared to the utilizations, right? So this accumulation of glucose 6-phosphate will see here, it will inhibit hexokinase, so that further glucose conversion should not happen, right? Further glucose should not happen. So this is well regulated. Inhibition, this is inhibited by same product, substrate level inhibition. That means this is, let's assume this is hexokinase, and hexokinase has a pocket in which this glucose 6-phosphate should attach, and once it attaches, it will inhibit, it will become inactive. It is like something is giving signal so that it will become inactive, right? But which is stimulating this? Once the glucose reach here, this hexokinase is activated and it converts, right? Or glucokinase is activated and it converts this. So if it is other cell, then hexokinase is activated. If it is in liver cells, then glucokinase is activated, right? The main regulatory mechanism is, as I told, transport of glucose from outside to inside is also regulated by insulin, right? But we only know that transfer of glucose is from outside to inside is regulated by insulin, but insulin has more function than that. Let's assume this is insulin. Once it has once the insulin attack here, the receptor will produce some signal, and that signals will come and activate this enzyme, right? And once this enzyme is activated, then rapid utilization of glucose will be there to run the glycolysis. That means insulin is not only acting to transport the glucose from outside to inside, it also helping the glucose to convert into the further reactions, right? So by activating the enzymes. What about glucokinase? How does it activated and how does it inhibited? That is very, very important, because this is a more specific enzyme which will be in liver, right? In liver, why? See also, in liver, nature has provided glucokinase, because when you have heavy meal, you want to high concentration of glucose will come. So that time glucokinase will be keep increasing their reaction velocity, like performance, keep increasing their performance, so that all maximum glucose can be utilized and or stored, right? Maximum glucose can be utilized or stored. So glucokinase is present in the liver. Are you getting it? So see the reaction. If you just imagine in liver, if hexokinase will be there, if you had heavy meals and just in liter concentration only, it become half. It will become like it will it will go there. In little concentration only it will become half saturated. If you increase little more only, it will become fully saturated. So even that whatever the glucose is coming, after that whatever the glucose will come, that will be not utilized, and it will return back to the circulations, and in blood you will find more amount of glucose, right? But nature is so smart that instead of hexokinase, liver has glucokinase. So if you have heavy meal also, so if even the concentration of glucose is high also, they can perform very well, and then they can utilize maximum, and only few amount of glucose is released into the blood, and in this way the blood glucose level is maintained, right? Now let's talk about this glucokinase, how it is regulated. I want to be very specific because it is very interesting and it is very unique mechanisms, and nature has taken care well of this glucokinase as compared to hexokinase. Before going to that, I want to highlight one more things. Out of hexokinase and glucokinase, which one will be have more spectrum of action? Spectrum of the action is from the when you have carbohydrate foods, then from the GIT the carbohydrate will broken down into monosaccharides, right? And the monosaccharides mainly into galactose, fructose, and glucose, right? So that three, galactose, fructose, and glucose. So there are three monosaccharides. So this hexokinase will be acting on all three, or glucokinase will be acting on all three. So broad of a spectrum, or a spectrum of action. Which one you think? I know you people have some different concept, but I want to highlight that most of the students will think that hexokinase will be having broad spectrum, like they can act on all the three monosaccharides, but glucokinase will be specific to glucose, right? But it's not true. It's not true. This glucokinase name is glucose. It resembles like glucose, but it's misnomer. Glucokinase is as broad a spectrum acting as hexokinase. Understood? So that means glucokinase can act on galactose, fructose, and glucose, all three, right? So that's the reason other name for glucokinase is, this is hexokinase D. Other name of glucokinase is hexokinase D, or hexokinase 4. Hexokinase 4. Are you getting this? So it's very, yeah. Now we'll see how this enzyme is regulated, right? So let's erase this part so that we'll have a space to, yeah. Now you see, do you know about headquarters of the cells, right? From where everything is controlled. So the headquarters is nucleus. And the nucleus, you know, from the gene, the genetic coding will be there. From the DNA replications and all, one protein is found, and that protein is formed, and it will be like stored inside the nucleus, right? And this is a special type of protein. All the protein will follow the same mechanism. So all this special type of protein which store this glucokinase. That means glucokinase will be stored in this nucleus. Okay, so this is a glucokinase which is stored in this nucleus with a capping of protein. So that is, this is called glucokinase regulatory protein. Glucokinase regulatory protein. So before the glucose come, it was stored in this nucleus. But how this activated, and it will come into the reactions? What do you think? See, once the glucose will come here, when the level of glucose, concentration of glucose will increase. See the magic. When the concentration is increased, the flow of the glucose will, glucose itself, this flow of the glucose will go here, and it will pull it back, and this activated, and it will come as a action, right? So who will activate? The concentration of glucose itself is the activation of this glucokinase, right? So this concentration of glucose activated, but who will inhibit? Will inhibit this. So for that, let's run this. We'll see it later. Who will inhibit this? We'll find it. So this is glucose 6-phosphate, which will further go in and form glucose 6-phosphate with the help of the phosphoglucose isomerase. Phosphoglucose isomerase. Okay, phosphoglucose isomerase. This will form 1, 2, 3, 4, 5, 6. This will form glucose. At the 6th position, what is there? Carbon. So this just isomerase. Glucose is also 6 carbon compound. Fructose is also 6 carbon compound. So this is just isomerase, and it is formed. Now it is called as fructose 6-phosphate. The good thing is that there is nothing to remember about this step. Okay, so but one thing you can remember, that is this fructose 6-phosphate, it will go, and excess production of glucose 6-phosphate can inhibit the glucokinase activity. Okay, so once the excess production will be there, that means that one in later phase, like once the glycolysis, enough amount of ATP is produced, and then the cells, okay, now don't run ATP like this in the late phase, and cells become lazy to run the forward directions, then there will be accumulation of fructose 6-phosphate, right? And then once they started the accumulation of fructose 6-phosphate, then they know that they will activate, they will inhibit this glucokinase, so that further oxidation of glucose should not occur, and glucose should release back into the bloodstream. Okay, so this is things. So in glycolysis, we are only focusing on those steps which are irreversible. Okay, so here we have talked more detail about this step. So this step is, if you remember, not remember, but you have to remember, just write few things about this one. So now what we will say is, this is again irreversible step. So I'll draw with red color. So this is irreversible step. And in this step, what will happen is, one more phosphate is attached. At which carbon? This is the origin, at carbon number 1. And then we'll see here, now it is, this product should be called as fructose 1,6-bis phosphate. Fructose 1,6-bis fructose 1,6-bis phosphate. Why bis? See, when the carbon number is not in adjacent side, like 1,6 is there, so we will use the term bis. But when there will be like just 5, 6, carbon number 5 and 6, then we will call bi or di. So fructose, if it is 5, 6, then 5, 6 biphosphate or diphosphate. Okay, so this is the concept. From where this extra phosphate has come? This extra phosphate has come from, I know you guys are smart, so this has come from ATP. So this ATP will convert into ADP, and this ADP is known as, that before ADP, this phosphate is released, and this phosphate will be attached at the first position, right? And the enzyme which which makes this is this happy enzyme, which is working well. So this enzyme is phosphofructokinase 1. Because there is some other enzyme for phosphofructokinase 2. So in books, if you see, there will be just writing some other, just write that PF phosphofructokinase. But you have to go more specific, phosphofructokinase 1. And as I told, this is the irreversible step. That means this enzyme should be well regulated. So let's discuss few more things about these enzymes. First things, when this enzyme should know, or when to run this, to convert this, right? So first activation, first how this enzyme is activated is, so first activation is, there is some pocket, there is some pocket in the enzymes which sense the level of ATP inside the cells. So if the level of ATP is more, so do you want to run these cycles, because this is cycle for producing ATP. So we don't want to run these cycles if the ATP is more. That means once this ATP level is increased inside the cell, that means this enzyme will get inhibited, right? And yeah, further direction should not be stopped. And now other product is which bind is citrate. So if the level of the citrate is more, that means what? TC is running well, more amount of TC is running, that means more amount of what is producing? Energy is producing, ATP is producing. So that means more amount of ATP is producing, means this will inhibit this, right? If citrate level is less, that means still ATP is required, right? So citrate running, TC cycle is running less if it is less, right? So level of citrate and level of ATP, if it is increased, it inhibits the fructose kinase, fructose kinase 1. But one more thing I want to highlight, I can't control myself, so just I love to mention one more thing that is very important actually. This fructose 6-phosphate, before this converting to this, few amount of fructose out of 100, just imagine 4 to 5% of fructose 6-phosphate converted into some other product, and this is reversible product. This can be reversible, okay? And this reversible product is called as, just see here, this reversible product is known as 1 phosphate is attached at position number 2 and 1 is attached at the position number 6, right? So this is called as fructose 2,6-bisphosphate, fructose 2,6-bisphosphate. So we can write here is fructose 2,6-bisphosphate, right? Why I'm talking more about this? Look this, the enzyme which convert this, they have dual role, because it is reversible, right? So it has dual role, and this enzyme is well regulated actually. That enzyme is well regulated. So let's talk about that enzyme which convert this. They have like phosphofructose kinase 2, phosphofructose kinase 2. But that is one component. Here is phosphatase also, phosphatase. So wherever you feel there is phosphatase, age means adding the phosphate, removing the phosphate. Kinase means adding the phosphate. So when this reaction is going forward and producing this component, that means kinase activity is going on, right? This enzyme is working, this part of enzyme is working, right? And when this phosphofructose phosphatase is working, that means this is taking the phosphate back, right? Removing the phosphate, and it is coming here, right? And this enzyme is well regulated by insulin and glucagon. See here again, this is very important to know. The role of insulin, I just want to highlight here. The role of insulin, how it affects in the internal steps of the glycolysis. If insulin is activated, it will release one substance, and that substance will remove phosphate from this group. Phosphate from here also and phosphate from here also. It will remove phosphate. It will remove. Once this enzyme will be dephosphorylated, then this will become activated. Kinase activity will increase, phosphatase activity will decrease. Remember the things. If this will remove phosphate from this phosphate, which insulin is doing, insulin is removing the phosphate, dephosphorylation from both sides. So when phosphate is removed from this enzymes, what will happen? This activity will increase and this activity will be inhibited. That means reactions will move in the forward direction. So when you have when you have enough amount of food, right? When you are in fed state, that means insulin level is high. That means it will dephosphorylize, right? So removing phosphate from this the hair, then that means PFK2 will be activated. That means this fructose 6-phosphate, 5% of the fructose phosphate will be converted into fructose 2,6-bisphosphate, right? So you just imagine from out of 100 molecules, just examples, out of 100 molecules, if 5 is producing this, 5 has converted into fructose 2,6-bisphosphate, and this fructose 6-phosphate will stimulate, right? Which activate and stimulate or activate this enzyme, like other rest 95 are remaining, right? So convert this rapidly, right? So this production of this enzyme, so production of this substrate will activate again this phosphofructokinase 1 and accelerate the reactions fastly, right? So this is very, very important, right? Or when you are in fasting stage, see other example, when you are in fasting stage, that time insulin level is low, then what level is high? Glucagon. When glucagon level is high, then this will produce one more substance, like you can tell like CMP and all, CMP, and this will ultimately act and aid the subtest, because from ATP it is converting to AMP or CMP, right? So there will be release of phosphate, and that phosphate will be attached here. That phosphate will get attached. So once this enzyme will be phosphorylized, when phosphate is attached, then kinase activity will decrease. Phosphatase activity will be stimulated. That means this product formation will reverse back, and this reaction will go like in reverse way, and you can glucose formation will be there, right? So when you have glucagon, glucose formation will be there, and glucose will release from this cell to the bloodstream to maintain the blood glucose level. So you are seeing how this enzyme is well regulated. This enzyme is very well regulated. I hope this much will be enough. Okay, and this don't forget that this is catalyzed by an Mn manganese or magnesium, these reactions. Okay, so up to this, from glucose to this state, this is called as, actually for the understanding purpose, this glycolysis is ordered in 3 to 4 stages. Some books it will be 3 stages, some book it is called 4 stages. So anyway, this stage you call as preparation stage, preparatory stage or preparatory stage or preparation stage, because in these molecules, up to these molecules, we can see this asymmetrical molecule of glucose is converted into symmetrical molecules. Asymmetric glucose converted into symmetric, because now the next step is splitting stage, in which we will see that this will break down exactly at the center, splitted, and you can see 3 carbon with phosphate, here also 3 carbon with phosphate. So next stage is splitting stage, and in splitting stage we'll see the splitting of glucose now, right? But till now, just recap it. We have invested 1 ATP. See, this glycolysis is meant for gain of ATP, not OTP, sorry, gain of ATP. But here in the first step itself, we are investing the ATP energy. We have invested. Here also we have invested ATP. So 2 ATP already have been used or invested. Till now there is no gain of ATP. But yes, of course, in the further steps we will see in detail how this ATP will be gained back, because at the end of the glycolysis at least we should gain few amount of ATP, then only it is like useful, right? Unless we are losing ATP, means what is the point of running this cycles inside our cells, right? Cell is not mad, right? So okay, we will continue this in the further lectures. Thank you. I hope if you have any doubt, please like, share and subscribe, and put a comment so that I can reply you in the comment.